Afternoon, everyone. Hope you've all been having a good couple of days here. This is going to be part 3 in our lesson on DNA and its structure and function. If you've not already watched part 1 and part 2, I strongly recommend you go back and view those first so that you have the necessary background for this lesson. If you are in my biology class, you probably should go on Blackboard and download the relevant handouts so that you have the information that you need for this. Now, at this point, I am assuming you already know the history of the DNA molecule, you know the basics of its structure, you know what a nucleotide is and how they join together. You're aware of complementary bases and how base pairs form to build the double helix. And you know about the various steps of DNA replication, initiation, elongation, and termination. And assuming all of that goes well, we should end up with identical copies of the original DNA molecule when all is said and done. Now, replication is obviously a really important thing we have to do. It's one of the two big functions that DNA has to execute. One of those, of course, is making sure that every new cell that we produce ends up with a complete copy of the genetic information, that it has all the data it requires to carry out all of its essential functions. But, of course, the really big function of DNA, the really important one, is going to be the question of exactly how does it do its job. Actually, before we get into that, let's back up for a second here. Last time we talked, I told you all about replication and the various steps of that. But one thing I wanted to finish out our discussion of replication with is a little bit about a thing that happens at the end there, about the process of termination. You might recall that one of the last things that happens in termination is that we add a little cap of sorts to the end of the DNA molecule called a telomere. T-E-L-O-M-E-R-E. And like I said last time, the telomeres are sort of like the aglets at the end of your shoelaces. They're there to basically protect the fragile ends of the DNA from external damage. The ends of the molecule are where the DNA is most vulnerable. It's where it's most likely to be affected by external forces, and therefore having the telomere there at either end is a buffer. It means that there's a portion of the molecule that can be damaged without having to necessarily harm the coding portions of the molecule. Now, here's the thing about telomeres, folks, is that they're built by enzymes called telomerases. T-E-L-O-M-E-R-A-S-E. Telomerases. Yeah, sorry, got lost in the word. But so your telomerases are the enzymes that assemble telomeres. But here's the thing. They're not equally good at doing that over time, not in all species. Some species are very, very good at regenerating their telomeres. Some are not. Most mammals are not. And what that means is that as the telomeres get damaged over time, that as they're subject to outside forces, they get worn down a little bit. They gradually shorten over time as you suffer more and more slings and arrows of chemical damage and physical damage and radiation damage and other things that can afflict a cell over its lifetime, which means that little by little, those protective telomeres get eaten away. But they're not equally repaired in all species, and we are not terribly good at fixing our telomeres. And what this means is that little bit by little bit, as we copy the DNA over time, as the DNA is replicated and new cells form, their telomeres are going to be a little shorter than they were before. And eventually, they're going to get a little shorter still. And eventually, the telomeres are effectively going to be worn away. And if that happens, now the DNA itself starts to get directly damaged. There's a name for this. This gradual damage to our DNA that happens over time as our telomeres get worn away. And it's actually not so much a name for this, but it's more a name for the effects that we get. Because as the DNA begins to become damaged, organisms are going to suffer characteristic effects that come with that because certain genes are located in certain spots in the DNA, and therefore once the DNA begins unraveling from the ends, certain genes are more likely to be affected. Therefore, certain functions and structures of the organism are more likely to suffer problems because of that. This gradual breakdown of structure over time as a result of the loss of telomeres is referred to as senescence. S-E-N-E-S-C-E. Senescence. You know by another name, aging. The effects that we associate with aging, the characteristic breakdown of bodily structure and function that you associate with getting older, is largely a consequence of the breakdown of telomeres, that our DNA is no longer as well protected as it used to be. It becomes damaged in certain characteristic ways, and therefore, as everybody gets older, they begin suffering similar effects as they age. It's not like some people get wrinkly and other people turn blue. You know, there are going to be certain things that happen to all of us as we get older because of the loss of our telomeres and because of certain genes being more likely to be damaged as a result of that. Now, it's not as simple as all that. That's not the only thing that is a driving factor in breakdown and change with age. Environmental exposure is a factor, nutrition is a factor, lots of things are a factor. But in as much as we can tell, telomere breakdown is the primary force that drives aging. Now, naturally, one thinks about this and is like, okay, can we rebuild the telomeres? Can we maybe, perhaps, find a way to slow down aging or to maybe even reverse it? The answer to that question appears right now in biology to be a qualified yes. Experiments with telomerase enzymes and endeavoring to rebuild telomeres in organisms that have suffered telomeric damage have demonstrated, at least in mice, that we can potentially extend their lifespans by up to 150%. Or by up to 150%, I should say. Now, not everything that we can do in a mouse can be equally well scaled up to a human being, but it strongly suggests that with some biological efforts, we could reasonably create anti-aging drugs. We could reasonably develop treatments that would reverse a lot of the potential damage of aging, or slow it down or prevent it from happening in the first place. Scientists who work in this relatively small area of research, anti-aging research, have suggested it is not unreasonable to think that with what we know about telomeres right now, you could potentially extend your lifespan to maybe as much as 200 years. And this is functional, healthy lifespan. This is not you being an old person for 170 of those years. This is you having a healthy body that is not showing the decline of age. And I'm going to tell you all right now, if you're an average college student watching this, within your lifetime, you are going to see anti-aging treatments. It's going to happen. There are enough people working on it. The proof of concept is there. We just have to find a way to scale it to humans. And when that happens, we as a society are going to have to do a whole lot of thinking about many, many, many aspects of our culture that make the basic assumption that everybody gets old and dies at a certain point. Whether that's everything from population growth rates to retirement age to lifetime appointments to certain political positions. All of this is stuff we're going to have to reconsider in a world in which you can reasonably expect people to remain healthy and functional well into their second century. We're not there yet, but make no mistake, it's going to happen. All right, though. Now, before we leave behind DNA replication too, one more thing I wanted to talk about is the differences in how replication works between prokaryotes and eukaryotes. Now, like I said in my last lecture, this process of replication is strongly conserved across evolutionary history. Virtually all living things go through more or less the same steps in their DNA replication process. The initiation, elongation, and termination that I laid out for you, the basic enzymes involved, that's pretty much the same in everything from E. coli bacteria to blue whales. Now, here's the thing. Not all cells are quite the same, though. Prokaryotes and eukaryotes have some very significant differences in the organization of their DNA. And therefore, there are going to be a few things that differ between prokaryotic and eukaryotic cells in how they engage in replication. Prokaryotes, as hopefully you remember, have all their genetic information in one place. They have a single ring-shaped bacterial chromosome that contains their entirety of their main DNA, plus, the little plasmids floating around here and there, which go through their own independent replication process. But your DNA is different. Your DNA is chromosomal. Your DNA is divided up into lots and lots of little individual packed up units called chromosomes. You as a human being have 46 chromosomes. Now, don't forget also the DNA is tightly compressed within those chromosomes. Chromosomes are basically a packaging system to cram more DNA into a smaller area. Because you have so much more DNA than a prokaryotes, we have some special considerations that come into play when it comes to replicating it. First and foremost, your DNA is not all one unit, therefore it can't be replicated all as one unit. Every chromosome has to go through its own replication process. So the steps of DNA replication that I laid out that are happening during the S phase of the cell cycle are happening individually to every one of your 46 chromosomes. There's not one continuous starting point to ending point for all of your DNA. Every chromosome goes through it on its own. Secondly, there's the fact that your chromosomes are a form of highly condensed DNA. In order to access it, we have to unravel them a little bit. So you're going to find that during the S phase of the cell cycle, the chromosomes relax. It's kind of like taking a spool of thread that's tightly wound and unwinding it so that it's, more loosely packed. It's a more loose ball of thread rather than like a tightly compressed ball of thread. That enables access for the enzymes to the various portions of the DNA that need to be copied out. And finally, there's the fact that you not only have more chromosomes, but you have a lot more DNA in general. Your entire genome is much, much, much larger than that of any bacterium. Just by way of contrast, the average bacterium has about 1,100 genes in its genome. You have about 25,000. Furthermore, you have a whole lot of parts of the DNA that don't code for much of anything. Our DNA has a whole lot of inactive regions called introns, I-N-T-R-O-N-S. Keep that word in mind. It's going to come up again later. The coding regions of DNA, the regions that contain your genes, are called exons, E-X-O-N-S. So exons are the coding regions. Introns are the non-coding regions. You have a lot of introns. Now, some of those introns are doing other things. Think about like the origin of replication, some of them like termination sites. Those are areas of the DNA that have a job. It's just not a job in containing genetic information. Others are stretches of what's sometimes called junk DNA, DNA that is not really coding for much of anything. And we're going to talk about why that is later. But because you have so many introns, plus the fact that you have so many more genes, you have much more DNA in general than any prokaryotic organism. If I took all the DNA in one human cell, unwound it from every chromosome, stretched it out end to end, it would be about 2 meters long. That's just in one single cell of your body. If I took all the DNA out of all your cells, stretched it out, and laid it end to end, it would cross our entire solar system multiple times. And that's in one human body. So you got a lot of DNA. Now, every cell is doing this independently, obviously. Not every cell is replicating at the same time. But you do have the issue of copying out that much DNA per cell. It's going to take longer, right? So we have a special adaptation to speed that up. Namely, that in eukaryotic DNA, you're going to find that there are multiple origins of replication. That the origin of replication is not just one spot in your DNA, that the origin of replication actually is a thing that you're going to see in multiple points along that DNA molecule simultaneously. To give you an idea what I'm looking at here, let me just draw you all a quick sketch that you saw before, that whenever I was showing you the origin of replication and how the DNA molecule unzips from the origin of replication, bidirectionally working towards either end. That's what I showed you with that before, right? That you see the boxy spot there, that's the origin of replication, that we're unzipping in both directions at the same time, working towards both ends of the DNA at once. That's what it looks like for a prokaryote. For you, it's going to look more like this. Bear with me a moment. This is more what your DNA is going to look like. That rather than having a single origin of replication, there are going to be multiple origins of replication along the length of the strand. As a result, the process of replication is going to be happening starting from multiple points on the DNA at the same time. That you're going to be having this whole process going on, not just from one spot, but from several spots, each one of them opening up a portion of the DNA molecule so that collectively they can all engage in this replication process faster. By copying the DNA from several points along its length simultaneously, you greatly accelerate the rate at which replication happens, which means that any given chromosome, all of your chromosomes are going through this process individually. But if I look at any given chromosome, I will find multiple origins of replication, multiple replication forks, multiple replisomes, all doing their jobs at the same time. That's all going to be happening at once. All those different origins of replication forks that I showed you, all going at the exact same time to speed up the process. And that's how we compensate for having so much more DNA than a prokaryote does, and still getting it all copied in a reasonable period of time. All right, replication. But we have not gotten to what is arguably the most important part of the DNA, the whole reason this molecule matters in the first place. Namely, how does it store data? What is the DNA doing that is letting it tell our cells what to do? What is happening in that sequence of A's, T's, C's, and G's that is instructing your cells on everything from how to make your eyes blue to how to create digestive enzymes? All the instructions for you are encoded in that DNA molecule. How? What is going on here? Now think back to our genetics history, folks. P.A. Levine, back in the 1920s, came to the conclusion that DNA could not possibly be the genetic molecule. And his reasoning was, it's too simple. He said it's only got 4 basic parts, right? A, T, C, and G. It's got 4 basic kinds of nucleotide. He said that is not enough complexity to encode for all the possible stuff that life needs to be able to do. You need a more complex operating alphabet than that. So for that reason, Levine said proteins must be the genetic molecule because proteins have 20 different amino acids. There, he said, there's enough complexity to begin coding for things. Now, obviously, we know today that Levine was wrong. The mistake he made was in assuming that the individual nucleotide, the individual A, T, C, or G, was the unit of information. That is not the base unit of genetic information. First off, what is DNA coding for? When I say, okay, it contains all the instructions on how to make you, you. All right, that's great, sure. But what does that actually mean? I told you way, way back in the beginning of this course that when you're thinking about DNA, DNA should be thought of as a recipe book for proteins. That is what DNA is. It is the master copy of all the instructions on how to build all the proteins that your body requires. By building all of your proteins, we can basically control everything because everything in your body is either made from a protein, is regulated by a regulation protein, or is a chemical reaction carried out with the help of a protein enzyme. So basically, even the non-protein molecules of your body are synthesized with protein instructions and controlled with protein regulation. So, by containing formulas for all the proteins in your body, I'm containing all the instructions for how you operate. How do I make your eyes blue? Well, I have a formula in my DNA that codes for the protein that is responsible for that blue pigmentation. How do I digest milk? Well, I synthesize a protein called lactase that breaks down milk sugar. So really, one way or another, we can control everything either by building the proteins that it's made from, building the enzymes that make the reaction happen, or building the regulation proteins, things like hormones, that control when and how processes take place. So by containing the recipes for all the proteins, I contain the entire formula for you. So that's what DNA is. DNA is the protein instructions. When we think of the units of DNA, we think of A's, T's, C's, and G's, those basic individual nucleotides. Those are not the unit of genetic information. What is a protein, after all, right, folks? A protein, you all know, is a sequence of amino acids, or it's multiple sequences of amino acids that you link together a bunch of amino acids to make a polypeptide, and then your protein consists of at least one polypeptide, possibly more than one polypeptide. So amino acid sequencing, that's what it's all about. If I want to tell my body how to build a protein, I need to tell it what amino acids it needs to do that, and I need to tell it what order they need to come in. Think way, way back to early part of this class. Think way back to when we talked about organic molecules. And I told you that every protein has a primary structure. And the primary structure of the protein is its exact amino acid sequence. That is, which amino acids go into that protein and what order they have to come in. Well, what DNA is actually doing is instructing your cells on the primary structure of all the proteins they need to build. It's telling your cells, okay, if you want to assemble that protein right there, you need this amino acid, and that amino acid, one after another. There are no proteins that are only 3 amino acids long, just, by way of example. Now, if I look at the actual genetic coding, that long string of letters, reading it as individual letters does not tell me the instructions. I have to read them in groups of 3. That was the mistake that Levine made. He thought the individual nucleotide letter was the unit of information. Nope, it's groups of 3 nucleotides. Every group of 3 nucleotides in the DNA, every sequence of like ATG or CCA or whatever else, every group of 3 nucleotides is called a codon. C-O-D-O-N. So each codon is 3 nucleotides. And each codon is a unique chemical code representing one specific amino acid. Every codon stands for one particular amino acid. So maybe the codon GAG stands for the amino acid valine, for example. That's just to pull an example out of the air. I'm not going to recite off the different codons for all the different 20 amino acids. You don't need to know them right now. If you take a biochem class, yeah, you're going to get into more detail, but I don't expect you to have that at the moment. But yeah, so every codon is the unique code for one particular amino acid. So if I am reading my DNA in the 5 to 3 direction, I can read down and I can say, okay, there is GAG. That's the sequence, actually that wasn't for valine, that's for glutamic acid, my apologies. So there's GAG, there's the sequence for glutamic acid. Let's say we have CTC. CTC is the sequence for valine, and so forth. So that every time I have 3 nucleotides, that represents one amino acid. Now, let's don't get confused here. I'm not saying that is an amino acid. A codon and an amino acid are not the same thing. Think of codons as words in the genetic language. That in our genetic language, we only have 4 letters in the alphabet, and every word is 3 letters long. And every 3-letter word in genetics represents one specific amino acid. So what I'm really doing is I am spelling out amino acid by amino acid the primary structure of the protein. So I can say, all right, I've got 7 codons in a row here. Those codons represent 7 different amino acids. So what my DNA is telling the cell is, okay, when I read those codons, this is the instructions on how to build that protein. That I need this amino acid, that one, in this particular sequence. And if I link those amino acids in that sequence, I will get the protein that DNA is coding for. So every unit of 3 nucleotides is the word in the genetic language for one specific amino acid, is instructing the cell, when you're building this protein, put that amino acid here. And then the next codon is the word for the next amino acid, and it tells the cell, here's the next one you need to attach, and so forth and so on until you get to the end of the chain. Now, you've all heard the word gene, G-E-N-E. The gene is the fundamental unit of genetic instruction. A gene is the complete set of instructions on how to build one polypeptide. So that's what a gene really is. You know, I say, okay, I got the gene for blue eyes. What that really means is that I have the gene in my DNA that contains the complete set of instructions for how to assemble the polypeptide that is responsible for the blue color of my eyes. That, okay, if I want to build that blue protein, I need these amino acids in that order. That is what the DNA is telling me, and that means, so when I say I have the blue eye gene, I'm really saying I have the set of instructions on how to assemble the blue protein. Now, you might note I said one polypeptide. The rule is one gene, one polypeptide, meaning that every gene codes for one particular amino acid chain. Let's remember that polypeptide and protein do not mean exactly the same thing. A polypeptide is a sequence of amino acids. Not every gene is an entire polypeptide, or rather, I should say not every protein is just one polypeptide. Some really simple proteins consist of a single polypeptide. Most are more complex. Whatever the codon language of DNA was discovered back in the 1950s, Francis Crick was involved in that after the publication of the structure of DNA. But the belief at the time was one gene, one protein. That every gene, every sequence start to finish of amino acids in the DNA was the chemical formula for one entire protein. We have since learned it is not that simple. We have since learned that it's really one gene is one polypeptide, one chain of amino acids. What that means is that if I have 3 different polypeptides in my finished protein, I need 3 different genes to build that entire protein. So in other words, it's not necessarily as simple as, oh, here's the gene for this. I might need 1, 2, 3 genes to do that if there are 3 different polypeptides involved. And so I have to synthesize all of those individually, and then they all have to come together at the quaternary level of protein structure to build the complete protein. So each gene codes for one amino acid sequence, that is one polypeptide. That polypeptide might or might not be a finished protein. And if it's not a finished protein, then basically we're going to have to decode another gene or another 2 genes or another 3 genes or whatever to build the finished protein. So your body's proteins are not necessarily all single gene units. A lot of times they're multiple gene units. And this is one reason, incidentally, why there's sometimes more than one form of a particular genetic disorder, for example. That you can have 3 different polypeptides that go into that protein, and if polypeptide 1 is messed up in some way, it causes that genetic disorder to happen in one way. Polypeptide 2 is messed up, it causes it to occur in a slightly different way. So, that's how I want you to think of your DNA. Strings of codons. Every codon, every unit of 3 letters, represents one amino acid. Spell up enough of those together, I build one gene. And the gene is the complete formula for one specific polypeptide that is instructing the cell on how to build one particular amino acid chain. And that amino acid chain might or might not be a complete protein molecule, depending on whether the protein in question has more than one polypeptide or not. All right, does that make some kind of sense? I'm asking like you can say something to me right now. You can't say something to me right now. You can send me questions if you have questions. But one last thing I wanted to say about that. I don't know if any of you stopped to do the math here, but let's think about some math for a moment. There are 3 letters per codon. There are 4 letters in the genetic alphabet. A, T, C, G, right? Any given codon can be any combination of those 4 letters with repetition allowed. So I could have a codon that says ACT, but I could have one that says AAA. Any possible group of 3 of those letters, including more than one copy of the same letter, is valid. 4 letters, groups of 3 with every possible group of 3 permitted and repetition permitted. I'll spare you the math. There are 64 possible combinations you can build. there are 64 different codons that can be assembled from the 4 letters of the genetic alphabet. Refresh my memory, how many amino acids are there in living things? 20, right? Yeah, there are 20 amino acids out there. There are 64 possible codons. In other words, my genetic language is more than complex enough to code for everything. Levine was wrong. Levine's mistake was thinking that individual nucleic acids, individual nucleotides, were the unit of information. Had we had to read it 1 unit at a time with 4 letters, yeah, it wouldn't have been enough. But reading it in groups of 3 letters, we have far more complexity than is required because we have only 20 things to code for and we have 64 possible ways to code for. So yeah, interpreted this way, the genetic language has far more complexity than is necessary to build all the amino acids for life. So you're probably wondering, what are all the extra codons for? There are 2 answers to that. One of them is that some of the codons have non-coding functions. Some of the codons do not code for amino acids. A few of them instead indicate, okay, here's the beginning of the gene, start reading here, and here's the end of the gene, stop reading there. We basically tell the cell, okay, here are the boundaries of the gene for whenever you're done decoding it and where you need to start decoding it from. We'll talk more about that when we talk about RNA in the next unit. Now, the other thing that's going on there is that there is actually a lot of redundancy in the genetic code. In other words, there is more than one codon for most amino acids. Most amino acids can be coded for in several ways. The sequences ATA, AAT, and ATT, all 3 of those codons represent the exact same amino acid. So there's considerable redundancy in the genetic code. Some amino acids only have 1 codon that represents them. Some have as many as 6. So there's more than one way to build any given amino acid. That redundancy is quite important because you'll notice that most of them are pretty similar to each other. Like AAT and ATA, just switch 2 letters around and, it's the same letters, right? That similarity is not a coincidence. Because what this basically means is that small mutations will not necessarily be an issue. That little mutations sometimes will make a small change in the DNA, but if that change is only by, say, 1 letter, it might not mean anything. It might not actually mess up the genetic code any. If I have a mutation that hits my DNA and it changes a codon that reads AAT to one that reads ATA, it still has not affected the amino acid that it's coding for. Therefore, having redundancy in the genetic code is an insurance policy against minor mutations. It means that small changes in the DNA are a lot less likely to lead to a change in the protein that is being coded for. And therefore, that means that the organism is more likely to be able to endure minor mutations without having harmful effects. All right, folks. So you have now heard what DNA is. You've now heard how it is built. You've now heard how it is copied. And you have now heard how it instructs the cells on what they need to do. That's the fundamental instructions in DNA. What you have not yet heard is how exactly we decode that information. That is, in other words, how do we go from letters in DNA to protein? I told you that, okay, this amino acid is coded for by that codon, but what does that actually mean? How do I go from a codon sequence written in DNA to an actual amino acid being linked up to another one to make a protein? That is going to involve another molecule. That's going to involve a thing called RNA. So, I know this has been a little shorter than most of my lessons, but we have hit a good stopping point. So I think at this juncture, we are going to close off our basic lesson on DNA. In my next couple of lessons, we're going to talk about gene expression. We're going to talk about how we actually decode the DNA and build proteins according to its instruction. We're going to talk about mutation, what happens when the genetic code is changed. And we're going to talk about heredity, that is, inheritance, how information is passed from one generation to the next. All right, but for now, I think you have all the information you need to know about the basic workings of DNA. Hope you all have a great afternoon. Talk to you later.